welcome, Dr. Kontaridis. Am I pronouncing it correctly?
Yes,
Great.
perfectly. Thank you.
Well, thank you. Thank you for joining us today. For the rest of us, our speaker today is Maria Kontaridis, PhD. Her topic is entitled From Pediatric Hypertrophy to Heart Failure. We will also address upcoming FDA regulatory updates. Academically, Dr. Kontaridis received her BA and BS from the University of Florida, her MS in pharmacology from Yale, her PhD from Yale, and she did her postdoctoral fellowship at Israel Deaconess and also at Yale. She is an assistant professor at Harvard, and we're very fortunate to have her tonight. Thank you again for joining us, and please, it's time for your presentation.
Wonderful. So I'm just going to share my screen. Thank you so much for the kind invitation. Let me see. Can you see that? Okay.
Yes.
Okay, excellent. So as I was listening a little bit to your introduction earlier, I want to just make very clear that I admire the clinical aspects of what you do for sure. I am a very basic research scientist, and I hope that some of the work that we do will lead to some translational aspects of what you do in the clinic down the road. So my research is largely focused on a lot of signal transduction, we call it, sort of the transition of signaling that happens from the extracellular matrix of the cell all the way down into the nucleus to tell the cell what to do. And a part of that is using a variety of tools to allow us to be able to do that. So my lab is focused primarily on a group of rare disorders called RASopathies. They're individually rare, but collectively they comprise the largest family of congenital heart defects in the world. And they include things like Noonan syndrome with multiple lentigines, Costello syndrome, and neurofibromatosis. And although you can appreciate that all of the genes that are consequent to the development of these disorders all reside within the same pathway, and this is the ERK-MAP kinase signaling cascade that's critical for proliferation or differentiation of cells, you can see that the kids that have these various disorders actually look quite different. So one of the major focuses of the lab have been to try to really understand what it is that makes these diseases similar, but more importantly, what distinguishes them and makes them different. So our lab has focused on one particular gene for a number of years called PTPN11, which is causal to both Noonan syndrome and Noonan syndrome with multiple lentigines. So some background on what SHP2 or PTPN11 is. It's a ubiquitously expressed protein with two N-terminal SH2 domains, a catalytic pocket, the PTP domain, and two tyrosyl phosphorylation sites at its C terminus. And it was really the elucidation of the crystal structure and the biochemistry that allowed us to really understand its biological function. And that is when it's inactive, it actually folds over and binds into its own catalytic pocket, rendering it inactive. When it binds or when a cell gets activated by a phosphotyrosine protein, it unfolds and therefore allows substrates to bind into the catalytic pocket to elicit downstream signaling. And the reason this is so important is because it's involved in virtually all receptor tyrosine kinase and cytokine signaling pathways, and is typically an upstream regulator of the RAS-MAP kinase cascade, but can also be a negative regulator of the AKT signaling cascade, which is involved in apoptosis. So when I looked at these two kids and thinking about their development of various diseases, what we discovered was that there were a lot of similarities, and that 50% of all cases of Noonan syndrome